Hello, everyone. Welcome to another episode of Deep Dive Knowledge Talk. Welcome, everyone, and thank you for joining us today. Today, we'll discuss how genetic algorithms can be used for molecular structure optimization. We'll cover how clustering techniques can improve efficiency and how reinforcement learning can further enhance performance. These findings have applications in a range of fields, including material science, drug discovery, and environmental science. Can you explain the significance of using genetic algorithms in the search for optimal molecular structures, particularly in the context of applications like drug discovery?
Genetic algorithms are like smart search parties looking for treasure in a vast forest. In the case of finding optimal molecular structures, which is important for drug discovery, these algorithms help scientists explore a huge range of possible molecular designs, much like how a search party would cover a large area to find hidden treasure. One of the key advantages of genetic algorithms is their ability to work in parallel, similar to how multiple search teams can look in different spots at the same time, making the search much faster. This is especially important in drug discovery, where time is critical and scientists want to quickly find the best molecules that can help treat diseases. However, searching for the best structures can lead to many unstable or less useful designs along the way, like a search party coming across many false alarms. To improve the efficiency of this process, clustering techniques are used, which group similar molecules together. This helps minimize the time spent exploring unproductive areas, allowing the algorithm to focus on more promising candidates. Overall, genetic algorithms play a significant role in making the search for new drugs faster and more effective by helping scientists find the best molecular structures while avoiding unnecessary detours.
How does the introduction of clustering in genetic algorithms enhance their efficiency in molecular structure optimization? And what challenges are associated with the selection frequencies between these clusters?
Introducing clustering in genetic algorithms is like organizing a big group of kids into smaller teams for a scavenger hunt. Each team focuses on a specific area, which helps them find treasures, or in this case, optimal molecular structures, more efficiently. Clustering allows the algorithm to manage and explore different groups of molecules, making it easier to identify the best candidates without wasting time on unproductive searches. This method enhances efficiency because by clustering, the algorithm can quickly assess which groups are doing well and which ones need more attention. It can then adjust how often it selects molecules from these different clusters based on their success, similar to a teacher giving more attention to teams that show promise while still encouraging the others to improve. However, there are challenges when it comes to selecting frequencies between these clusters. If one cluster consistently performs well, it might dominate the selection process, which would mean the algorithm might miss out on valuable discoveries from other clusters. This is like letting one team in the scavenger hunt pick all the best hiding spots, while the other teams might have also found good treasures if given a chance. To balance this, the algorithm uses specific parameters to reward or penalize clusters based on their performance. If a cluster does well, it gets a higher chance of being selected again. But if it does poorly, it receives a stronger penalty, encouraging the algorithm to explore other clusters that might eventually yield better results. This system helps ensure that all teams have a fair shot at finding treasures, and it can help the overall search be more fruitful.
Can you elaborate on the specific parameters defined in the study, such as MFavOvrAll-A and Select-D, and explain how they contribute to managing the exploration-exploitation trade-off in the context of cluster selection?
The parameters defined in the study, such as MFavOvrAll-A and Select-D, play important roles in managing the balance between exploration and exploitation in the optimization of molecular structures using genetic algorithms. MFavOvrAll-A acts like a gold star system for clusters that produce the best molecular structures overall. When a cluster finds the absolute best structure, this parameter increases the chances that this cluster will be selected again in future searches. This is akin to rewarding a student for good grades, encouraging them to keep performing well. This parameter promotes exploitation, meaning it focuses on the successful clusters that have already shown they can find excellent solutions. On the other hand, Select-D is more like a timeout for clusters that get picked too often. If a cluster is selected too many times, Select-D reduces its selection probability, ensuring that it doesn't dominate the search process. This helps to encourage exploration of other clusters that might have been overlooked. Imagine if a teacher only called on the same student every time. Eventually, other students wouldn't get a chance to answer. Select-D helps to make sure all clusters get a fair shot, even those that might initially seem less promising. The balance between these two parameters is crucial. If MFavOvrAll-A is set too high, the algorithm might only focus on a few successful clusters and ignore others, leading to stagnation in finding new solutions. Conversely, if Select-D is emphasized too much, the algorithm could end up randomly selecting clusters without considering their past performance, which may result in less effective searches. In summary, MFavOvrAll-A and Select-D work together to ensure that the genetic algorithm is efficient. They promote a healthy mix of exploiting known good solutions while still exploring new possibilities, making the search for optimal molecular structures both thorough and effective.
What methodology was used to optimize the parameters defined in the study, and what were the key findings regarding the effectiveness of these parameters in enhancing the performance of the genetic algorithm?
The methodology used to optimize the parameters defined in the study involved a systematic approach to testing different combinations of parameters through simulations. The researchers utilized a grid search method to explore a range of possible values for 4 key parameters: MFavOvrAll-A, MFavClustB, MFavClustC, and Select-D. Each of these parameters influences how the algorithm selects clusters based on their historical performance in producing low-energy molecular structures. To optimize these parameters, the researchers first modeled the performance of each cluster by calculating the average and standard deviation of molecular energies produced from multiple evolutionary algorithm runs. This modeling helped to create a Gaussian distribution for the values generated by each cluster, facilitating quicker evaluations of parameter combinations. The optimization process involved running simulations where groups of 5 clusters were selected multiple times, with each group undergoing a series of evolutionary algorithm iterations. The lowest energy values produced were tracked, and the probabilities of selecting these clusters in the future were adjusted based on the defined parameters. This iterative process allowed the researchers to determine which combinations of parameters yielded the best results, specifically targeting lower energy configurations. Key findings from this study indicated that a careful balance between the parameters was necessary for enhancing the performance of the genetic algorithm. The combination of MFavOvrAll-A and Select-D was particularly crucial because it illustrated the need to balance the benefit of exploiting successful clusters, while still allowing for exploration of potentially fruitful but underperforming clusters. The results showed that optimizing these parameters led to significant improvements in finding optimal molecular structures, demonstrating the effectiveness of the dynamic probability-based cluster selection approach compared to traditional methods. Overall, the study highlighted that the right parameter settings could not only decrease computational time and resources, but also improve the quality of the solutions found in the search for low-energy molecular configurations.
How did the proposed reinforcement learning-based method for cluster selection outperform the unclustered evolutionary algorithm in the context of quinoline-like structure searches? What specific advantages were observed?
The proposed reinforcement learning-based method for cluster selection outperformed the unclustered evolutionary algorithm in the search for quinoline-like structures due to several specific advantages. First, this method effectively utilized clustering to group similar molecules, allowing for a more focused search. By applying techniques like Oganov's fingerprint function and hierarchical agglomerative clustering, the algorithm could better assess the performance of different clusters. This focused approach helped reduce the noise from unstable intermediate structures, making it easier to identify the most promising candidate. Second, the dynamic selection of clusters based on previous performance significantly enhanced the algorithm's efficiency. Parameters like MFavOvrAll-A, which rewards clusters for producing the best structure overall, and Select-D, which penalizes over-selected clusters, created a system that balanced exploration and exploitation. This meant that while the algorithm could exploit successful clusters, it also ensured that other clusters had the opportunity to contribute, preventing stagnation in the search process. The results from the study showed that this reinforcement learning approach produced more consistent and effective outcomes compared to the unclustered method. The unclustered evolutionary algorithm lacked the structured feedback mechanism provided by the cluster selection process, leading to less optimal results in finding low-energy molecular configuration. Overall, the combination of clustering and adaptive parameter tuning in the reinforcement learning-based method led to enhanced performance, demonstrating that this approach could yield superior results in the search for optimal molecular structure.
In what ways could the findings of this study be applied to future research or practical applications, particularly in fields beyond molecular structure optimization?
The findings of this study could have significant applications in various fields beyond just molecular structure optimization. Here are a few potential areas where the methodologies and results could be beneficial. 1. Material science. The techniques developed for clustering and optimizing molecular structures could be applied to understand the morphology of physical defects in materials. By using similar algorithms to identify optimal arrangements of atoms or molecules, researchers could improve the design of new materials with desired properties, such as increased strength or resistance to wear. 2. Drug discovery. The success of the genetic algorithms in finding low-energy configurations can be directly applied to drug discovery processes. The ability to efficiently search for optimal molecular structures could lead to the rapid identification of new drug candidates, ultimately speeding up the development of pharmaceuticals and improving treatment options. 3. Biology and biochemistry. Similar methods could be utilized to study complex biological structures, such as proteins and enzymes. Understanding how these structures fold and function can lead to advancements in biotechnology and therapeutic interventions. 4. Computational chemistry. The reinforcement learning-based approach can be adapted for other computational chemistry tasks, such as predicting reaction pathways or simulating molecular dynamics. This could enhance the understanding of chemical processes and lead to the discovery of new chemical reactions. 5. Artificial intelligence and machine learning. The principles of dynamic cluster selection and reinforcement learning could be extended to other fields in AI, such as optimization problems in logistics, finance, or even game playing, where efficient search strategies are critical. 6. Environmental science. The optimization techniques could be applied to model and predict the behavior of pollutants or other environmental factors, helping in the design of better strategies for environmental protection and remediation. By further refining these methods, researchers can potentially extend their applicability to larger and more complex structures, enhancing the breadth of research scenarios where these techniques can be beneficial.
Thank you all for joining us today. We've explored how genetic algorithms leverage a search party analogy to tackle the vastness of molecular structure optimization. Clustering enhances efficiency by grouping similar molecules, guiding algorithms towards promising candidates. Balancing exploitation and exploration through parameters like MFavOvrAll-A and Select-D is crucial. Reinforcement learning further optimizes cluster selection, surpassing unclustered algorithms. Beyond molecular structure optimization, these findings hold potential in various fields, including material science, drug discovery, biology, computational chemistry, AI, and environmental science. We encourage you to explore further and apply these concepts to your work. Thank you again, and have a wonderful day.